Welcome to Office Hour, week number 9, about the second part of photosynthesis. We discussed that photosynthesis contains two parts, the light-dependent reaction that depends on light in order to make high-energy electrons, and the second part will be the light-independent reaction that does not really depend on light. It can happen during daytime or nighttime. It really doesn't matter, independent from the light. So let's go and review a little bit how the two reactions relate to each other. The photosynthesis had the first one, and in office hour number 8, week number 8, we discussed that contains the light-dependent reaction. And this time we had the water coming through the xylem from the soil, and the water split into oxygen and hydrogen ion and electron. The electron and the hydrogen ion was picked up by NADPH, NADP plus, and it's going to get reduced, picking up the electron. So it's going to be NADPH. And the second end product of the light-dependent reaction was the ATP. We call it when inorganic ADP plus inorganic phosphate with the help of ATP synthase produced ATP. So these are the two end products that they move over to the light-independent reaction. The second part is light-independent reaction. Good. So here we are actually still in the chloroplast, outer membrane, inner membrane, and that happened in the thylakoid membrane. Oops. This was in the thylakoid membrane, recall this. Not really the ADP, this was outside. Okay, something like this in the thylakoid, and we are in the stroma now. Inside, still in the chloroplast. In the stroma, we have a molecule called the ribulose biphosphate, RUBP. You do not really need to know those names, but I just say it's coming from the ribulose biphosphate. We start with, for counting wise and to understand the carbon situation, we will start with 3 RUBPs, and each RUBP contains 5 carbon atoms. So altogether, this is 15 carbon atoms we start with. Here comes the carbon dioxide through the atmosphere, stoma, and going to combine with the RUBP, ribulose biphosphate, with the help of an enzyme. It's the Rubisco. This facilitates as the inorganic carbon in the form of carbon dioxide binds with an organic molecule. This is called the carbon fixation. This is the carbon fixation, step 1 in the light-independent reaction. During carbon fixation is going to turn into a 6-carbon molecule, 3 of them, but because they are very unstable, these molecules, the long 6-carbon ones, they are going to split into we will have 3 6-carbon molecules, and it will split into those long 6-carbon molecule into phosphoglyceric acid, 3-phosphoglyceric acid. These are 3 carbons. So altogether 18, 3 times 3 is 9, 3 times 6. So we still have 18 carbons in this reaction, like 3 times 6 is 18, but 3 carbon phosphoglyceric acid and 3 phosphoglyceric acid is 6, but each of them contains 3 carbons. So altogether still we have 18 carbon. This phosphoglyceric acid is going to get the high energy electrons at that time. So this part, these molecules are going to get reduced. Where does the high energy electron come from? Well, we have the NADPH, and this is coming, okay, around here. So this NADPH is going to turn into NADP+, so it's going to get oxidized. In the cellular respiration, we will get again into the redox reactions that they cannot happen on their own. There's no oxidation without reduction and vice versa. So this NADPH is going to get oxidized to turn NADP+. High energy electrons, these are really the important ones. What made the high energy electrons? Well, the energy from the light got transferred, and it's the chlorophylls electrons got excited, high energy, thing back, light dependent reaction, and it's going to get the 3-phosphoglyceric acid is going to get reduced to glycerol aldehyde 3-phosphate. So this is, I just change it to black color, glycerolaldehyde 3-phosphate. Again, we are going to have 6 molecules. Let's divide the 6 up to 5 to 1. And 5 to 1 is important because this 5 well, not that fast. So this first one by itself will go out, and if the circle happens twice, In this case, the two glyceraldehyde 3-phosphate will combine together to form the sugar, the glucose. But this cycle has to happen twice. So that step was the reduction step. Reduction of the PGA molecule, and it's going to be the G3P molecule. And the last step, this 5 molecules of G3P, that it's 15 altogether now, because this glyceraldehyde phosphate contains 3 carbons. 3 times 5 is 15, and this is going to be regenerated. This RUBP, this will be the regeneration and step. We left this out, the ATP, for that reduction reaction. ATP helps, and for that regeneration, ATP helps. So the ATP production in the light-dependent reaction is going to be used in this cycle. That cycle is also called the Calvin cycle, or, the Calvin cycle. This Calvin cycle is how the carbon dioxide, the inorganic carbon dioxide, is going to be fixed. Through reduction is going to form the glyceraldehyde phosphate, and regenerating the molecule that can fix the carbon dioxide with the help of Rubisco. I guess that's all for the light independent reaction. Have a good one.